Welcome back to this next video, and in this video we are going to talk about Mendel and his laws. The full name of Mendel, it was Johann Mendel, and he was given this title, the Gregor, when he joined the monastery of the St. Thomas. Mendel was born in 1822 and he died in 1884. If you talk about Mendel, he was a scientist. He was a friar of the Augustinian order, and he remained the abbot of the St. Thomas Abbey Church. Now, Mendel, he is known as the father of genetics. So let us discuss what this genetics mean. If you talk about genetics, it is actually the study of pattern of inheritance in individuals and in population. Now, most of the time when you talk about the genetics, it is usually taken as the genetics of the individual. You talk about the pattern of inheritance in individual, but there is a specific field of genetics which is called as the population genetics. And in population genetics, the pattern of inheritance is usually studied in the population. When you talk about the pattern of inheritance, there should be something responsible for the inheritance. And that's something which is responsible for inheritance, that is known as a gene. Now, what is a gene? By 2020, it is still a debatable thing how to define a gene, and that is mostly dependent on the tradition you come from. For example, if you are a molecular biologist, your definition of the gene will be sequence of the DNA that encodes functional RNA. That is how the molecular biologists, they define a gene. And when you talk about an RNA in a particular cell, you will be having different kind of the RNAs. You will be having the messenger RNA from which the proteins they are made. You can have the you will have the transfer RNA, which are actually the RNA responsible which are taking their part in the translation process. The ribosomal RNA, which are the structural units of the ribosomes, the machinery of the translation. We talk about the small RNA, which are known as the miRNA. They are very important in the gene regulation. And there are a lot of the RNAs in the cell which are known as the TUFs, the transcripts of the unknown function. So if they are RNA in nature, that means there will be a gene for them. A gene is transcribing them, but as we do not know their function, we are calling them as the transcripts of the unknown function. Now, that doesn't mean that they do not have a function, but as we do not know their function, therefore we call them as the transcripts of unknown function. If you are a geneticist, your definition of the gene, that will be different than the molecular biologist. The geneticists, they define the gene as a heritable unit of function, not necessarily a nucleic acid. Like the molecular biologists, they say that the gene, that will be a sequence of the DNA. That means you will be talking about the nucleic acids. But when you talk about the geneticists, they say that a heritable unit of function, but that heritable unit of function do not need to be a nucleic acid. Like prions, they are the heritable unit of function, and they are not nucleic acid in nature. They are protein. Still, they function as a unit of heredity. So depending on the tradition you come from, your definition can be sequence of the DNA that is called in RNA. If you are a geneticist, you can say a heritable unit of function, which is not necessarily a nucleic acid. Now, all genetic differences, they are heritable, but not all things that are heritable are genetic differences. Now, you inherit a lot of things. That doesn't mean that all of the things that you inherit, they are the genetic differences. For example, your family name, genetics has nothing to do with your family name. If you talk about your religion, if you talk about the wealth you get in inheritance, this particular inheritance has nothing to do with the genetic differences. Therefore, we say that all genetic differences are heritable, but not all things that are heritable are genetic differences. And these are just some of the example of the things that you inherit, but they do not have any genetics as its background. Now, these genetic differences, they are heritable. There are certain genetic differences that are heritable, but are not passed through generations. And we call these genetic differences as the somatic mutations. This means that if you talk about, for example, the cancer, that will be a good example of the somatic mutations, because when a cancer cell divide by the process of the mitosis, it do transfer it genetic material into the daughter cells, but those daughter cells that remain in that particular individual, they are not transferred to the next generation. So these somatic mutations, like the cancer, and they remain in that particular individual and they are not transferred to the next generation. Now, if you talk about the life of the Mendel a little bit, the father of the Mendel, he was a poor farmer. And Mendel, he was not a robust kind of the guy. He was physically weak. So his father decided that as he is not fit for farming, he should send him to become a teacher. He should send him to studies so that he can become a teacher. So when he was sent for the studies, he joined the St. Thomas Church. And when he joined the St. Thomas Church, the then abbot, his name was the Abbot Nab, he made him a teacher. So Abbot Nab, he decided to give Mendel to teach to the lower classes. He was a very good teacher, but the problem was that some of the parents complained that as he do not have any teaching credentials, he should not be teaching our kids. So the Abbot Nab decided to send Mendel to the University of Vienna in 1851 so that he can get some teaching credentials. At the University of Vienna, he was under the influence of three important teachers. One was the Unger, a plant taxonomist. The other one was Doppler, who was a physicist, and the Eddinghausen, who was a mathematician. And specifically, the Unger and the Eddinghausen, they have a very important role when you talk about the laws of the Mendel. Mendel has got a problem, and that was known as the test anxiety at that particular time. When he spent 2 years at the University of Vienna, he took the exam and he failed the exam because he was having this certain problem which is known as the test anxiety. You know everything, but when you sit on the exam, you cannot reproduce what you know. So he was a test anxiety, he failed the test, and he came back to the monastery of the St. Thomas. So when he came back, the abbot, he made him the TA, the teaching substitute or the teaching assistant. And the teaching assistant, as it do not require any kind of the credential, so he can continue teaching to the students. So he was made TA, and he was given the responsibility to work on the blending inheritance to make good plants. What I mean by good plant is that during the blending inheritance, you have got two parents. Both of the parents have got some good characters, and when you combine them, you can actually get the good characters of both parents in a single offspring. So he was given this task to work on the blending inheritance. The reason was that the most of the land around the Brno, where this particular church is present, so most of the land that was owned by the church, and the church was interested in blending to make good plants, and in making good plants, they can make some good money. So when he started working on the blending inheritance, the known things at that particular time to Mendel was the blending inheritance, that you can combine two parents with good traits, you can expect the good characters of both parents in a single offspring. The unknown at that particular time, chromosome was not known, mitosis, meiosis, the ploidy, the DNA, nothing was known at that particular time. And this shows how genius Mendel was. Like, not knowing all of these things, he actually described the mitosis, the meiosis, the inheritance in a very good fashion. Now, most of the scientists, many scientists, they were also working on the blending inheritance at that particular time. But what was the reason of the success of the Mendel? Now, the most important thing that made him successful was that he chose his experimental material wisely. So the most important thing when you are going for some kind of the experiments, that you have to choose your models very wisely. If you choose them wisely, you can interpret the results very good, and you can work with them in a fashion that you can get to somewhere. So he chose his experimental material very wisely. The experimental model he chose, the garden pea, botanically that is known as the Pisum sativum. One of the important characteristics of it was that it can be easily grown in the experimental gardens or pots in the greenhouse. So you can actually grow them very easily. That means if you can grow them very easily, you can do a lot of experiments with them and you can interpret your results very easily. Another important thing was that these pea plants, they contain both the males and the female organs. Like the males, they have got the anthers which produce the pollen grains, which was the male, you can say male organs, and the female organ, the ovary, which was producing the eggs. Now the petals of the flower, they close down very tightly, enforcing a self-fertilization system. The importance of this was that if the petals, they are closing very tightly and if they are posing a self-fertilization system, you can actually have true breeding varieties. And if you have true breeding varieties, you can actually determine the pattern of inheritance in these true breeding varieties. But the most important thing of his success was that he focused on single traits at a time, as compared to the other scientists who focused on many traits simultaneously, making the results very complex. At the time, he was actually focusing on a single trait, and if you are focusing on a single trait, you can actually deduce your result very easily. But if you are focusing on multiple traits at a time, which many scientists did at that particular time, the result will be very complex and you will not be able to reach anywhere. So this was the most important point which led to the success of the Mendel. Now, when Mendel started his work, he actually selected the pure breeding varieties. The pure breeding variety means that if you have got a particular plant and its seeds, they are round in shape. If this particular plant, if this particular seed is a pure breeding, that means that if you grow these seeds, in the next generation, you will be getting the round seeds. If you grow that generation, in the next generation, again, you will be getting the round seeds. So as many times you grow them, all of the time you will be getting the round seeds. So that is what is meant by the pure breeding varieties. Now, in the technical terms, if you talk about them, you can say that these particular seeds, they are homozygous for the round trait. Both of the alleles, they are of the same nature. Mendel selected 7 traits to study. One of the trait, that was the shape of the seed, and it come in 2 varieties. Now the trait, that was the shape of the seed, but the varieties, they were 2. The seed shape, it was round, and in some cases it was wrinkled. So you can actually have the shape of the seed as round or wrinkled. If you talk about the seed color, it can be yellow or it can be green. If you talk about the flower color, it can be pink, it can be white. If you talk about the shape of the pod, it can be inflated or it can be pinched. If you talk about the color of the pod, it can be yellow or it can be green. If you talk about the flower position, that can be apical or that can be terminal. And the last one that he focused on was the stem length. So the stem, it can be long, it can be short. Famously, that are known as the tall and the dwarf plants. So these were the 7 traits that Mendel focused on, and he got the pure breeding varieties for all of these traits, and he used them during their experimentation. Now, as I've told you that Mendel started his work on the monohybrid crosses. Now, the mono means single, the hybrid means that the trait you are focusing on, the gene you are focusing on, that have got 2 different alleles, and the cross means that 2 parents, they are actually crossing or they are combining their genetic information. From the monohybrid crosses, he actually deduced the principle of the dominance and the principle of the segregation. We will talk about them. Now, in one of his experiments, in the monohybrid crosses, he crossed a pure breeding, as I've told you, we call them as the homozygous. He crossed a pure breeding tall plant to a pure breeding dwarf plant. And he did that by the cross fertilization. As I've told you that the pea plant, they were actually self-fertilizing themselves. But what Mendel did was that he went for the cross fertilization. So how he achieved this cross fertilization? Say for example, these are the two plants. If you want to cross them, what Mendel did was that if this is a plant, this is the ovary, and it also have the anthers, the female organ and the male organs. So what Mendel did was that he removed the anther from this particular plant, from this particular flower, and that was emasculated, and that means that the male organ that was removed, that means that you have stopped the self-fertilization. What he did with the other plant, that he collected the anthers, he removed the anther, and he collected the pollen grains from this particular plant, and he used the pollen grains of this particular flower to fertilize the ovary of this particular flower. So it was a cross fertilization, because the ovary that was from a different flower, the pollen grains that was from a different flower, therefore we call it is the pollen grains. And he did this in both directions. What I mean by the both direction is that he crossed the tall male with the dwarf female, and he crossed the dwarf male with the tall female. So he did this in both direction. Like for example, if this is the tall male and if this is the dwarf female, what you will be getting is that you will be using the pollen grains of the tall flower to fertilize the ovary of the dwarf female. In another case, what you will be having is that you will be, if this is the dwarf male, you will be taking the pollen grains of the dwarf male and you will be using that to fertilize the ovary of the tall female. So he did that in both directions. So if you cross them, so as we are talking about the pure breeding varieties, that means that the tall that will be in the capital T and in the capital T form. The dwarf that will be in the small t and in the small t form. What Mendel did was that if you talk about these genotypes, the capital T and the capital T, I'll tell you in the study, I'll tell you that later on that he deduced that the genes, they are actually coming in pairs. And if they are coming in pairs, that means that it have two alleles, and they are actually making a pair. So capital T, that will actually be a pair of the alleles. Similarly, the dwarf, the small t and the small t, and he when he crossed them, when he crossed the pure breeding tall plant with the pure breeding dwarf plant, this particular tall plant is making a single kind of the gamete, which is known as the small t. This dwarf is making a single kind of the gamete, which is known as the small t. So when he crossed them, what he get in the F1 generation was that all of the plants, all of the offspring, they were tall in nature. The dwarf character, that was gone. What he did next was that he went for the self-fertilization. The tall plant he got in the F1 and the capital T and the small t, he crossed that with another plant of the F1 having the same genotype, the capital T and the small t. If you talk about these plants, this particular plant can make two kind of the gametes. It can make gametes having a capital T. It can make a gamete having the small t. This particular plant, it can make two kind of the gametes, a capital T and the small t. If you cross them, when he crossed them, what he get was that this is the capital T, this is the small t. I have a detailed video on the Punnett Square, how the Punnett Square work, how the gametes they are formed. I would share the link in the description. So if you talk about the Punnett Square, the outside of the Punnett Square, like this particular side and this particular, the top of the Punnett Square, they are actually showing you the gametes of the parents. And inside of the Punnett Square, you are actually having the fertilization, the fertilized product. That means the offsprings. So if you talk about this parent, it can make two kind of the gametes, and we are showing the gametes over here. If you talk about the other parent, we are showing the gametes over here. When he crossed them, this particular gamete, it can combine with this particular gamete, giving you an offspring with the genotype capital T and the capital T. If you cross this particular gamete from this parent and this particular gamete from this parent, you will be actually having the capital T and the small t, and that will be a tall offspring. If you cross this capital T with this small t, you will be getting the capital T and the small t, and if you talk about this gamete and you combine that with this gamete, you'll be actually having the small t and the small t. So in the F2 generation, what he got was that 3 of them, they were tall, and 1 of them, there was dwarf. So for every 4 plants, what he got was that there was actually a ratio of 3:1. If you talk about these particular plants, the F2 generation with a ratio of 3 tall to 1 dwarf, if you talk about the genotype for the tall, that was actually in 2 forms, the homozygous tall and the heterozygous tall. This homozygous tall, it was only present in 1, its genotypic ratio was 1, but the genotypic ratio of the heterozygous tall plant, that was 2, and the total phenotypic ratio, that was 3. That out of 4, 3 of them, they were tall. If you talk about the dwarf one, that was represented by this genotype, small t and the small t, its genotypic ratio was 1. That means out of 4, only 1 was dwarf, showing its phenotypic ratio as the 1. Now, what Mendel did next was that he the F2 plants, they were self-fertilized to produce an F3 generation. Mendel took it a step forward and he made an F3 generation, but the F3 generation, that was actually made by the self-fertilization, and that was not made by the cross-fertilization. What he deduced from the F3 generation was that all of the dwarfs that he got in the F2 generation, they produced only dwarf offsprings, demonstrating that they were homozygous for the T allele. That means that all of the offspring, they were producing a single kind of the allele, and when the allele combined with each other, all of the time you are actually getting the dwarf plants. So the condition will be like this, like one of the parent can be small t, again the other parent, that will be small t and the small t. This parent can make a single kind of the gamete carrying the small t. This parent can make a single kind of the gamete carrying the small t. So if you cross them, all of the time you will be getting the small t and the small t. That means that all of these offsprings, they will be dwarf in nature. One thing to keep in mind is that what Mendel discovered over here was that the in the F1 generation is the all of the offspring, they were tall in nature. So Mendel said that the tall trait or the tall phenotype, that is a dominant one, and the dwarf phenotype, that is a recessive one. Because the in the F1 generation, all of the plants, they were tall in nature, although you are having the alleles for the dwarf as well. But Mendel said that as the tall is representing itself even in the presence of the dwarf alleles, so the tall, that is a dominant trait, and the dwarf, that is a recessive trait. The F2 plants, the F2 tall plants, when they were self-fertilized, they comprised of 2 categories. Approximately 1/3 of them, they produced only tall plants. That means that if you take the tall plant of the F2 generation, that means if they are homozygous in nature, that means if you cross them, all of the time, all of the offsprings, they will be tall in nature. Now, 2/3 of them, 2/3 of the F2 tall plants, they produce a mixture of the tall and the dwarf offspring, thereby representing them as heterozygous in nature. So if the capital T and capital T comes, that will be producing a tall offspring, but if the small t combined with the small t, that will be actually giving you the dwarf plant as well. So 2/3 of them, 2/3 of the F2 tall plants, they produced a mixture of the tall and the dwarf offspring. Therefore, he deduced that the offspring, they were heterozygous in nature. Now, from these observations, Mendel deduced the principle of the dominance, and the principle of the dominance states that in a heterozygote, like for example, if you got this heterozygote, one allele may conceal the presence of another. That is what we see when the condition that is heterozygous. Now, the capital T that is representing itself and that is present with another capital T, but it is also expressing itself when it is present with the small t. So what he said that in a heterozygote, one allele, this particular allele, that may conceal the presence of another, and he established this particular principle of the dominance for all of the 7 traits that he selected. Another important principle, which is famously known as the first law of Mendel, that is the principle of segregation. This says that each member of a gene pair has an equal probability of being in a gamete. So what Mendel said was that the genes, they are also in pair. And if you talk about the pea plant, as he's talking about the pea plant, so the gene pair means that the pea plant, they are diploid in nature. So he must be given the credit to establish the diploid nature of the pea plant. So what he said in the principle of the segregation, that each member of a gene pair that has an equal probability of being in a gamete, that means that during the gamete formation, the members of these gene pair, they are going to get separated from each other. But a gamete gets only one of them, that when the members of the pair, they get separate from each other during the gamete formation, a gamete can only get one of the member from the pair. It actually cannot have both of them. It will be having only one of them from the pair. We'll continue this discussion in the next video.